Well, let's
Cool.
get started because these are scientist speed sessions. We are just, carving out a little piece of people's days and maybe a little bit of their lunch break. So let's get started with our very first scientist speed session with Matt Eddy. Dr. Eddy is researching sensory proteins and how they help us perceive our world. He's going to tell us a little bit about his research, and then it's all open for questions. This is an Ask Me Anything style kind of session. So if you have questions about Dr. Eddy's research, put them in the chat. If you have questions about anything, what Dr. Eddy likes to eat for breakfast, put it in the chat. If you just are here to listen, that's also fantastic. Thank you for joining us. And just so that I have said it, I want you to all know we have tons more virtual sessions planned, and lots of videos and games online at our open house kind of central location. I've added that to the chat for all of you to check out after this session. So take it away, Dr. Eddy.
All right. Well, good morning, good afternoon, everybody. I'm Matt Eddy. I'm an assistant professor at the University of Florida, Department of Chemistry, and also affiliated faculty of the National MagLab, which brought me here to this open house session today. So I'm going to go ahead and share my screen here with everybody, and I'll just start to go through a few slides to kind of introduce a little bit about what we study, and then it's up to you where we go from there. And feel free to ask me about the research or about myself, my job, anything that you're curious about. So let's go ahead and I'll just pull this up real quickly. So our group is interested in the general question about how we perceive our world and how we interact with it. And we know that this takes place using sensory proteins, specialized sensory proteins on every cell, not just in humans, but in humans they play very special roles, and we're interested in understanding the roles and interested in what they look like. So we're all already familiar with many of these proteins. So for example, how we perceive light, this is mediated by a specific sensory protein. And an amazing fact that you could take with you is that it's so sensitive that we can detect as few as just a few photons of light. So just a few particles of light are enough for us to be able to see. And I think that's just absolutely amazing because of these sensory proteins, and they can amplify the signal that we get from light to much bigger signals that go to our brain and that allow us to perceive light. So other kinds of sensory proteins that I'd say make our lives much more enjoyable or fun are ones that we interact with different kinds of foods and beverages. So we have, of course, chocolate featured here on the left, and we have coffee in the middle and tea on the right. And one thing that's interesting about all of these beverages is that they all contain different chemicals. So you can see at the bottom here, there are chemicals. Don't worry if you don't necessarily recognize all the details here, but just know that they all look kind of similar. And it turns out that these compounds are present in each one of these beverages or in chocolate, and they interact with a sensory protein in our brain. In fact, this is one that we study in my lab, which we call the caffeine receptor. So if you drink beverages that contain caffeine, this is the protein that is responsible for mediating those effects of caffeine. And another interesting point is that it's also a validated target for treating Parkinson's disease, and it's currently being explored in cancer co-immunotherapies as well. So another relationship that I want to emphasize is that many of these sensory proteins play very important roles in human health and disease. There are other sensory proteins within the same family of proteins that we study, and those are proteins that can alter our moods, our brain chemistry, and our health. That includes opioids. As we all are aware, the opioid epidemic is a very, very difficult problem that we're still struggling with to solve. And it includes also cannabinoids. So humans have two different cannabinoid sensory proteins, CB1 and CB2, and they're some of the most highly expressed sensory proteins in your brain. So besides, the fact that this is in the news a lot, of course, and the discussion around cannabinoids, these proteins are also being explored for treating rheumatoid arthritis, for treating obesity and other metabolic diseases as well. So again, we have these interesting relationships between these sensory proteins and human health. And so not surprisingly, these proteins that I'm talking about here belong to a much bigger family called G protein coupled receptors, GPCRs. They're receptors, and they sense many kinds of stimuli, and we're already familiar with a lot of these. So I mentioned the cannabinoid and opioid receptors, but dopamine receptors are GPCRs. Serotonin receptors that not only mediate effects of small molecules in your brain, but also external stimuli as well. Adenosine receptors, so I've talked about with coffee and caffeine. So anybody who has been stressed out or who has ever experienced a rush of adrenaline is also familiar with these proteins because they mediate the fight or flight mechanism. Chemokine receptors, so our immune system is loaded with sensory proteins that are trying to detect friendly molecules from foe molecules and aid in the immune system recognition of foreign bodies. They're also targets for a lot of new therapies. So some of the most recently developed anti-HIV compounds target chemokine receptors. Taste receptors. So everyone has two different kinds of taste receptors. These are sweet and savory, or umami. These are also GPCRs within the same family of proteins. And olfactory receptors. So humans have, in fact, over 400 different kinds of olfactory receptors that smell actually trillions of molecules. These receptors are not only expressed in your nasal cavity, though, but we find them in your intestines, in your guts, in your brains. What are they doing there? They are sensing other molecules. So in fact, your microbiome, all of the other organisms in your microbiome, also produce small molecules that interact with GPCRs. So that's one way that they interact with your cells, so to speak, is through these receptors. As I mentioned, they're expressed throughout the entire body, and they're involved in almost every aspect of human health and physiology. So we talked about vision, but they're also heavily expressed in the heart. Allergies, anyone suffering from allergies, those responses are mediated by histamine receptors, which belong in the same family of proteins, and we also study some of these. They're heavily expressed in our brain and our central nervous system, and they mediate our sensation to pain, and they likely mediate consciousness itself. As I mentioned, they're highly found everywhere in our immune system, in metabolic processes, and et cetera. So everything that you can think of, every physiological process typically involves one or more of these same proteins. And also, not surprisingly, because of this very important physiological role, they are big drug targets by labs in academia and labs in industry. So over 1/3 of all approved, all FDA-approved drugs target this family of proteins. And this is just a chart here. I won't go through the whole thing, but you can see there's fortunately an increasing trend, not only in the total number of drugs that have been approved by the FDA over the years, but you can see the dark blue bars here for each year, the number of drugs approved that target just this family of proteins that we study. So quite a bit, very important drug targets. And I'll just go through a couple of other numbers for you. So as I mentioned, over a third of all FDA-approved drugs target GPCRs. But what's really important, one thing to keep in mind, humans have actually 826 unique receptors, unique GPCRs. So far, we've only successfully targeted about 40 to 50 of them. And so there's huge potential in the future for new kinds of drugs that might successfully target GPCRs. And you can see here, here's a sort of list of some commercial names for different drugs that target GPCRs, and probably some of us are taking some of these right now, in fact. So where does the MagLab come in, and my role in studying these proteins? Well, in order to recognize all these different kinds of molecules, these are very dynamic machines. These are little molecular machines that have to change their shape in order to interact with other things, whether that would be a photon of light, whether that would be an opioid, caffeine, other proteins in your body, and etc. And in order to actually identify and recognize those different molecules, they have to change their shape. And we're interested in studying that dynamic behavior. So how these proteins actually change their three-dimensional shape to recognize and interact with other molecules. And I'll summarize it with this quote, from the Nobel laureate Richard Feynman, who'd said, everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And so we want to understand that jiggling and wiggling that's going on that enables all of these amazing functions to take place. That's sort of pictured here on the right. You can see this sort of cartoon about this motion of the protein moving back and forth. And this is obviously this is kind of a simplified cartoon, but it gives you a sense of what we are trying to visualize. We want to visualize that motion that allows these proteins to bind and interact with other things. And this is sort of a very general idea of how we do that, is that we make those proteins ourselves, actually. And so we make lots of them to study them. And we include what are called stable isotopes, which I'd be happy to tell you more about as well, that allow us to do experiments using really, really big magnets. So this is where the MagLab comes in to play, because they have some of the best facilities in the world that allow us to study these phenomenal proteins. And we can take our special samples, and we use our very, very high magnetic fields to do nuclear magnetic resonance experiments that allow us to visualize this jiggling and wiggling of atoms. And then we spend a lot of time also working very hard to understand that data and to process the data and allow us to visualize what interesting relationships we can find between these small molecules, drugs, and proteins by studying this jiggling and wiggling. So maybe I'll pause here and see if there are some questions, and then if there are more, I can show some more slides after that too.
That's fantastic. So my first question is, at the MagLab, of course, as you mentioned, we're all about big, powerful magnets. What kind of magnetic fields do you leverage to do this research?
That's a great question. So to get a sense of how strong these magnets are, so our Earth's magnetic field is something like quantitatively like half a Gauss. And the magnets that we work with in the MagLab are many orders of magnitude stronger than the Earth's magnetic field. So something like 10,000, 20,000 or more times stronger than the Earth's magnetic field. So they are very specialized instruments that we can't buy it off the shelf necessarily, and they're very unique. So the MagLab has not only instruments that are commercially available, meaning that there are large industrial companies that do produce this equipment, but they also have equipment that was built entirely in-house, so by really great engineers and staff at the MagLab who are experts about magnetic technologies that allow us to have these very strong magnetic fields. So very specialized equipment. They're so strong that if you had something magnetic in hand and you slowly approach the magnetic field, you'll feel the pull on that. So if you were to take a wrench or anything else magnetic and get too close, the pull would be so strong it would just slip right out of your hand, and it would give somebody a heart attack probably. But they're very, very strong.
There's a question in the chat that actually reminds that's a great question about nuclear magnetic resonance. You know, the what's up with this word nuclear, right? Is it is it like nuclear power? Like the question is, does NMR use nuclear? And maybe you could explain that, but also explain to people like what is NMR and what are these big magnets actually showing you, right? Why do you need a magnet that's that much stronger than the Earth's magnetic field? To do what? To see the proteins? Talk a little bit more about that.
Yeah, those are really good questions. So what we do using NMR is actually using the same very similar technologies that you would use if you go to a hospital and get an MRI. So when you get an MRI, the MRI stands for magnetic resonance imaging. And in fact, there are MRI instruments that are part of the MagLab suite of instrumentation. When you get an MRI, they don't say nuclear magnetic resonance imaging. They typically just say magnetic resonance imaging. It's using the same technology. And the reason for that is because there can be a kind of a stigma around the idea of nuclear. What does this mean? I think it can make people a little nervous. They might think about nuclear radiation, for example, and it turns out it's actually quite a bit different technology. And so we're not using ionizing radiation like you would have if you were to get a PET scan, for example, or another, or a nuclear fusion or nuclear fission reactor. That's a very sort of different kind of physics than what is going on here. Instead, what we are doing is, I'll explain in a second where the nuclear comes in. The nuclear just means that we are actually studying the nuclei, the actual nucleus, the core of the atom. So we are not exploding the atom, we are not fusing the atom with other atoms, but we are studying the atoms themselves. And so that's kind of where the nuclear part comes in, is because what we interact with in our proteins are actually the nuclei of all the atoms that make up our protein. Or if we are studying, if you get an image done of your brain or of your arm, they're looking at the nuclei, typically of water, in your brain or some other part of your body. So that's kind of where the nuclear part comes in, is that we are observing the nuclei of the atoms. And why are we observing the nuclei of the atoms? It's because they have a very, very special property called spin. And it turns out that they are magnetic. They can interact with a magnetic field like a bar magnet. So you can think about all of these little nuclei and atoms behaving like little bar magnets. And if you put them in a magnetic field like a bar magnet, they will kind of align with that magnetic field. Or if we change the magnetic field, we rotate it or we turn it, they're going to move with the magnetic field too. So they have this property that they act like little bar magnets. And that's why we focus on the cores of the atoms, the nuclei themselves. And we use the magnetic fields to manipulate these individual, these nuclei that behave like bar magnets. And we do that whether we want to generate an image of your brain or in a way kind of give us an image of your protein sample that we're studying.
That's that wiggling and jiggling Feynman was talking about, huh, Dr. Eddy?
Exactly.
I love
Exactly.
that. Great question. Don't forget, add your questions to the chat right now if you have questions for Dr. Eddy. Carlos, I know you have a couple questions.
I did. I want to go back to that question that you mentioned, that you said, or the magnet that you mentioned, that if you get too close, it'll pull that wrench right out of your hand. How many Teslas is that magnet?
Oh, that's a great question. So there's a spectrum of different instruments at the magnet lab, like there is across the world. And so there are instruments that are somewhere like a few Tesla, all the way up to the best of my knowledge, a world record 35 Tesla. So the most powerful magnetic field available in the world for these kinds of experiments is at the magnet lab. And it covers a pretty broad range. And keeping in mind that 1 Tesla is equivalent to 10,000 Gauss, and then the Earth's magnetic field is about half a Gauss. So that gives you a sense of how strong I think these magnets are. So even the weakest magnets, so to speak, are already really powerful.
That's right. That's like core MagLab brand, right? Massive magnets. Dr. Eddy, I wanted to ask you a question. When did you kind of first hone in on sensory proteins? I mean, this is a very interesting, I'm enjoying my sensory skills right now with a beautiful cup of coffee, but what was your inspiration for this field of study? Did you always know that you wanted to do this? Talk a little bit about your path toward this area of science.
Oh, that's a great question. My path has changed quite a bit and still changes as I learn and as I hear about what other researchers are doing and think about what is the most interesting and exciting research we can do. I got interested in these proteins when I was training during my PhD. I wasn't studying these proteins, this class of proteins, as a PhD trainee. I was using magnetic resonance in my studies. In fact, nuclear magnetic resonance to study solid kinds of samples. So these sensory proteins live in the cell membrane. This is basically a layer of fat that encloses all of our cells and encloses all cells in general. And these proteins live in this layer of fat that encloses the cell. And they're not the only molecules or proteins in this neighborhood. I was studying some other proteins that were in the same kind of environment. And I started to read more about this area when I was thinking about what did I want to do next. So after I complete my PhD, what would I like to do next? And I thought about different kinds of ways to move forward. I was pretty excited to have an opportunity to get some additional scientific training and learn about something a little bit new, something different that could complement my PhD training. And so I started reading more and more about these sensory proteins. And at the time, there were a number of really pivotal breakthroughs that were occurring. So scientists have known about these sensory proteins for a long time, and they've been very challenging for us to study and for us to learn more about. And at the time when I was getting interested in them, there were a number of breakthroughs that had been achieved, including breakthroughs by Nobel laureates that had been achieved that permitted that allowed us to even study them at all. And it was, I think, for me, it was seeing these breakthroughs that gave me the confidence and the vision that we could study them and we could learn something interesting about them. And maybe I could contribute to the knowledge that we learn about these proteins and their behavior and how they work. And maybe that knowledge could be helpful for designing new drugs or understanding better human biology, and so on. And so that was what got me interested in these, was just exploring broadly different scientific topics and finding one that I thought, wow, that's sounds really cool. I'd like to learn more about it. And that's what I did.
The rest is history, huh?
Yeah, and we're still studying these proteins. Of course, they're very exciting for us. And the more that we learn, it feels like we uncover more questions as we go along.
I'm glad you just said that right now, because one of the questions I always have students asking me is, as a scientist, are you worried that you're going to run out of questions? And I'm like, I don't think that's a possibility.
Yeah, there's no retirement in sight for scientists that I'm aware of. I think in part because, oftentimes what we do is driven by an initial question that we have, like, how does this protein, how does our immune cell recognize a foreign cell, recognize a bacterium or something else? And in the process of addressing that question, we generate 10 new questions that just open up new possibilities for us to explore. And maybe some of those questions are, how can we, not only asking deeper to get to a deeper level of understanding, but asking, how can we take this information and can we redesign a cell? Could we redesign a protein? Could we engineer a cell to make it better able to detect foreign invaders? In fact, that's what's being done right now in cancer immunotherapies, is how can we re-engineer immune cells so that they're better able to recognize cancer cells and kill them. So I'm definitely not worried about a shortage of questions anytime soon.
Okay, good, because I've got some off-topic questions for you. Are you ready?
Oh, good. Yeah.
One of the questions that my students always ask me is, what's your favorite movie or book?
That's a great question. And that's difficult to really...
It's hard to narrow it down to just one sometimes.
I can think of so many. When I think about books growing up that made very impactful impressions on me, A Wizard of Earthsea by Ursula Le Guin is one. Many books by Madeleine L'Engle, of course, A Wrinkle in Time. To Kill a Mockingbird made a very lasting impression on me. Oh man, so many others.
What about movie? You gave us a couple books. Pick a movie now.
Movies that made strong impressions on me,
or just
that
your
I
favorite.
Just enjoyed. Well, okay, I'll give you a range of a few movies. So I'd say a movie that made a strong impression on me before was probably Gattaca. And the idea that we can have a whole discussion about this, that we can shape our futures from a personal will or determination to do that. In terms of just fun movies, silly, I'd have to say something like Talladega Nights is pretty funny. Tropic Thunder, actually there was an article about that recently, those are some guilty pleasures.
That is quite all right. I've got plenty of movies that I will be afraid to admit that I own. So
Yeah.
I completely understand that. How about, I see you drinking your water bottle. What's your favorite in-lab snack?
Ooh, well, to UFHNS, we do not eat in my lab.
Understood.
But saying that too, yeah, no eating in the lab. But outside of the lab snack, near the lab, would be, that's a good question. Well, there's a lot of coffee around here.
Coffee fuels science, right?
No, yeah, so I drink quite a bit of coffee. My colleagues and students do too. You know, a good yogurt's always, I feel like, a good snack. You know, has like protein and it's kind of filling. So definitely a fan. You know, I like Fahe yogurt or yogurt with, you know, plain yogurt and honey is good too. And I think, you know, on the healthier side, you know, yogurt, nuts, fruit, those are always, you know, good things to have. You know,
And on the non-healthy?
The more guilty pleasure side, you know, well, we whenever we have, well, before the pandemic, as a group, we had group meeting in person, of course, and hopefully we will again before not too long. You know, I always get donuts for everybody. And so we're across the street. Well, we have within walking distance access to Dunkin' Donuts, Krispy Kreme, and then I think a little bit further along there's another like more mom and pop donut place. And yeah, donuts are a lot of fun. Most people like donuts.
I have a question about food. Since your area of study is about these sensory proteins, do you think that you and your research group are particularly sensitive to like tastes, like more than me, like because you study it?
That's a good question. I wouldn't necessarily
Or
relate that to more refined tastes per se, but I mean, it's kind of fun for making jokes about like, oh yeah, we study the caffeine receptor or something like that. But I would say it does give us a little more appreciation for the biology. I don't know if that's, you know, when we're eating a donut, that's, you know, probably not really going through most people's heads, but yeah, it kind of gives you an awareness that, you know, what we're studying is, I mean, we encounter it every day in our lives. It's not something really esoteric or that's, you know, something far off in the future. That, you know, what we are interested in, we're surrounded by external stimuli of different sorts, and that we, you know, what we study is directly related to our life experiences. You know, I asked this question, for example, we have a really outstanding collaborator at the National Institutes of Health, who's a medicinal chemist and is a world expert on the adenosine receptors, the caffeine receptors. And I've had a chance to talk to him a number of times in person. And, you know, I asked him, I said, what is your advice? So caffeine, good, bad, you know, what do you say? You know, what is the advice you tell your family about caffeine and health effects? And so we can have those kinds of conversations, and they're directly relevant to what we study in the lab.
Yeah, I'm sure that comes in handy just out and about and in the world. I just have a question about what what is it like being a scientist that you think is different from how most people think of what it's like to be a scientist? You know, like when you see scientists
Yeah,
on TV or movies or something, and you're like, oh, it's not like that at all. Like, what do you think, you know, how would you describe what doing science is actually like?
Yeah. How do you describe the disappointment they don't have a giant layer in a volcano?
how do you know?
How many times have you said eureka in your career, for example? Or
Um, in jest, I don't know, maybe half a dozen times or something. But well, yeah, it's interesting because there are moments of surprise. There are moments of discovery, but there's nobody there necessarily who's going to tell you that it is a moment of discovery. It's, I mean, you're coming to that realization, and um, it's kind of like I think putting in a lot of. I mean, scientists work very, very hard. They're, you know, many of the scientists I know are really, really hard workers, um, and they're people who are very focused and who are driven by, you know, ultimately some kind of um, urge to understand our world, and feeling a need to um, learn about how things work, and or learn about themselves, or maybe they're driven by a desire to improve our world, or improve human health, or diagnostics, or um, energy, and solve problems that are important to society. Um, but yeah, it's, I'd say on a day-to-day basis, you know, as a PhD student, you're very focused on, you're very like laser focused on um, your area of study, and you have, you know, some probably project or projects related to that area of study, and are training and learning, and at the same time trying to take the skills that you learn, and and then to apply them, to discover something new, or to come to a realization that nobody else in the world has come to before. So I think if I were to kind of romanticize about it a little bit, you know, there are moments where it's kind of cool to have the realization that you may be the only person, you may be the first person in the world to see something, or to develop an understanding, or there may be only very few people in the world that also have a similar perspective or unique insight into um, a specific problem. And that can be a fun, uh, I think can feel exciting. Of course, the next part is communicating those discoveries, and I think scientists could probably do a much better job at that part, communicating with other people their discoveries, and why they're important, and why people should support science. Um, so, I mean, my what I do now on a day-to-day basis is different than what I did as a PhD student, or as a postdoc. So now I'm a scientist, but also have a managerial role too. So it's kind of like operating a small business. You know, we have a budget. We have to worry about making sure that we utilize resources appropriately. You know, I work with students. I have to make sure that they're trained and that they fulfill their obligations in order to successfully complete their PhD programs and to get jobs afterward. You know, and teach as well. And I teach and do research and all of these things. So in my current role, I have to kind of wear a lot of different hats. So um, if you like that aspect, you might like this kind of role as well, because it's something new every day, and there are lots of, yeah, there's always more than enough to do.
I love that. I love that inspiration. We have one more question in the chat that I want to ask, and we'll probably leave it there at the end of this question. But is there a specific inaccuracy in a show or a movie, sort of a scientific inaccuracy that just drives you crazy? Something that
Oh,
was just completely.
yeah. I mean,
Matt,
well, what can I
I'll buy you a
say?
second to think about it, because one thing that always drives me crazy every time I watch a movie and they go, this spaceship material isn't even on the periodic table. And I'm just like, that's not
Yeah.
how it works. That's my personal pet peeve, but I'll let you answer with yours, because that one always drives me crazy.
Yeah, that's a great question. You know, I think years ago I had to kind of just let it go, because it happens so often, that it almost, you know, even movies that strive to be somewhat scientifically accurate, you know, Interstellar, for example, is a movie that uh, they really genuinely worked hard, I think, to try to provide some scientific accuracy. I mean, that has limitations, of course. In the end, you have to produce something that appeals to a wide audience. So I think certain performance choices are made to do that too. If I were to pick one specific thing, and I would say it's maybe an inaccuracy because, like, I wish it were true, is anytime, and this happens in TV and in movies, when, for example, like a crime related, crime solving related series or movie, you know, there's an image that is taken of, you know, and within that image, let's say, like half a mile away is like a tiny little car or something like that, and then the, you know, the researchers come to a conclusion that, you know, oh, that's the little car, that was like the getaway vehicle that the robbers took, like, let's try to enhance the image to get the detail, and they like, you know, and they blow it up, and then, you know, somebody presses a couple of buttons, and then magically it's like crystal clear, like what the make and model of the car is and the license plate number. And like that thing, that's definitely inaccurate. But I'd say that bothers me because I wish it were true, because I wish in many of our experiments we're really often limited by like fundamental physical limitations. It's like whatever we do, we're not going to get past a certain barrier, and that could be in microscopy, that could be in, you know, NMR spectroscopy, et cetera. That, you know, we have some kind of fundamental limitation we have to deal with, and so I just wish that we could press a few buttons and, you know, say enhance, and then our data is 10 times better. That, yeah, so that would maybe be one thing
Well,
that would
maybe
drive me crazy.
maybe someone watching today will be working on that for you, Dr. Eddy, and that'll be their contribution to the future. Anyone out there, that's a need. Scientists have it. Everybody has that need. So somebody out there, be inspired and work on that. All right, I think we're going to have to leave it there today. This speed session has been so much fun. We could just keep talking and asking and answering questions all day long, but we have to end sometime. And Dr. Eddy, this was such a wonderful presentation about our senses and the important way that they impact our health and all the new ways that could be coming soon from research groups like yours. I appreciate everyone who joined us today. Thank you so much for coming to this Open House Scientist Speed session, and check out our website for more Scientist Speed sessions later this week and into next week about all sorts of different scientific topics, because magnets can do all sorts of things. Special thank you to the National Science Foundation and the state of Florida for funding the National High Magnetic Field Laboratory, and of course, Dr. Eddy and the University of Florida. Thank you so much today.
Oh, thank you very much. If anyone has additional questions, I see there's a few more that popped up in the chat. Just feel free to shoot me an email. I'm really happy to chat with anybody. You can find my profile on the MagLab website, I believe, and I also have my own group's website. And I'm happy to chat more.
Fantastic. So get in touch with Dr. Eddy for more information. Thank you so much for everyone for joining
Thank
us.
you.
Take care. Bye now.